So, next topic is antileprotic drugs. That is, those agents which are used in the treatment of the leprosy. Now, what exactly is a leprosy? Leprosy, it is also called as the Hansen's disease. It is a chronic disease and caused by the bacteria Mycobacterium leprae or Mycobacterium lepromatosis. So these are the two causative organisms, that is the Mycobacterium leprae and the Mycobacterium lepromatosis, which cause the leprosy. The primary sign of the leprosy is the skin lesions. If the leprosy in initial stage, if it is not treated, then it causes permanent damage to skin, nerves, limbs, and eyes. Same as antimycobacterial, same as the tuberculosis. The Mycobacterium leprae, it is also spread from person to person through respiratory droplets. So now we will see the agents which are used in the treatment of the leprosy. So the first class is sulfones. So the diaryl sulfones present the major class of agents used to treat leprosy. Now, which one is the diaryl sulfones? So the first agent which is belonging to the sulfones are dapsone. It contains diaryl sulfone group. This dapsone was first introduced in the treatment of leprosy in year 1943. So this is the structure of the dapsone which is in front of you. And if we nomenclate the structure, how to nomenclate the structure? Now, first we will consider this group. Either one of the two group is the aniline. So we'll start from the aniline. So how will number the aniline from the nitrogen? So 1, 2, 3, 4. So 4 dash 4 amino phenyl sulfonyl aniline. So that is how we nomenclate the dapsone. Now dapsone is a diamino diphenyl sulfone and it is water insoluble agent and is having very, it is a weak basic compound having pKa is approximately 1. Because of the lack of solubility, it causes GI irritation, gastrointestinal irritation. Despite of the lack of solubility, the drug is effectively or efficiently absorbed from the GI tract. Although the dapsone is bound to plasma protein approximately 70% and it is distributed throughout the body. So the dapsone is what? Dapsone is a diamino diphenyl sulfone and it is having very weak base. It is a very weak basic compound and having pK is approximately 1. And dapsone is less soluble and despite of the less solubility, it is absorbed from the GI tract. Regarding the mechanism of action of dapsone, the mechanism of action is same as sulfonamide. That is, this drug, the dapsone drug, inhibit the enzyme dihydropteroate synthetase. The enzyme is necessary in the conversion of PABA to the folic acid. So the bacteria or the organism will not get the folic acid for the synthesis of the bacterial cell wall. So this is what the mechanism of action of the dapsone. That is, this drug inhibit the enzyme dihydroteroate synthetase. The enzyme is involved in the conversion of PABA to folic acid. So once the enzyme is inhibited, then the conversion of PABA to folic acid is also get inhibited. Regarding the SR studies, the several derivatives of dapsone have been prepared in an attempt to increase the activity. The isosteric replacement of one benzene ring result in the formation of thiazole sulfone. Okay, so in dapsone, if one benzene ring is replaced, then the resulting compound is thiazole sulfone. Although this thiazole sulfone is still active, but it is less effective than the dapsone. Substitution on the aromatic ring to produce acetosulfone, which reduced activity while increasing water solubility and decreasing GI irritation. So on the dapsone, on the aromatic ring of the dapsone, if we substitute the aromatic ring with the acetyl group, then it results in the formation of the acetosulfone. This compound is highly water soluble and there is a less GI irritation. A successful substitution consists of adding methane sulfonate to dapsone to give sulfoxone sodium. Okay, so if we substitute methane sulfonate to the dapsone, it will give sulfone sodium. This sulfoxone sodium is a water soluble form of dapsone and it is hydrolyzed in vivo to produce dapsone. Sulfoxone sodium is used in individuals who are unable to tolerate dapsone because of the GI irritation, but it must be used in dose threefold that of dapsone because of insufficient metabolism to the dapsone. So what actually is the sulfoxone sodium? That is, if we substitute the dapsone with the methane sulfonate group, then it results in the formation of the sulfoxone sodium. This sulfoxone sodium is water soluble and it is hydrolyzed in vivo to produce dapsone. So those patients who not tolerate the gastrointestinal irritation, then for those patients sulfoxone sodium is given. Once sulfoxone sodium is consumed, then inside the body it is get converted into the dapsone. But the drawback of the sulfoxone sodium is what? That is, it is given three times more of the dose of the dapsone because of the insufficient metabolism to the dapsone. So to meet the demand of the dapsone, the sulfoxone sodium is given three times more of the dose of the dapsone. Then it gives the effect of the dapsone in the body. The chemical modification of dapsone derivatives continues to be pursued with the intent of finding newer agent useful for the treatment of resistant strains of the Mycobacterium leprae. So still some chemical modifications are going on for the to treat the organisms Mycobacterium leprae. So these are the metabolic products of the dapsone. So what we have studied regarding the SCR, that if we replace one benzene ring, it results in the formation of the thiasulfone. So this is the drug thiasulfone. Then if we add the acetyl group to the dapsone, it results in the formation of the acetosulfone. And just we have studied about the sulfoxone sodium. So inside of the body, this sulfoxone sodium is get converted into the dapsone. So these all are the metabolic product of the dapsone. Then again, few further more metabolic products are given. The major metabolic product of dapsone results from N-acetylation in the liver by N-acetyl transferase. So these are not metabolic products. These are what? The structural activity modifications. Okay, these structures are the structural modifications of the dapsones. This is what the metabolic products make corrections. Okay, so the major metabolic product of dapsone result from N-acetylation in the liver by N-acetyl transferase. Dapsone is also hydroxylated to the hydroxylamine derivative. The metabolic reactions are catalyzed by CYP3A4 enzyme in the liver. Neither of these compounds possess significant leprostatic activity, although N-acetyl diaminodiphenylsulfone can be deacetylated back to the dapsone. The product found in the urine consists of small amount of dapsone. The metabolites N-acetyl diaminodiphenylsulfone and N-hydroxy-diaminodiphenylsulfone and the glucuronide and sulfate of each of these substances. So these are the metabolic products of the dapsone. But none of these compounds possess significant leprostatic activity as compared with the dapsone. Okay. Now the next drug is clofazimine. The next drug is clofazimine. So this is the structure of the clofazimine, and here I have mentioned the numbering for the better understanding of the IPC nomenclature. So we'll start with the N dash. Then there are two similar groups. Hence, we first consider those similar groups because also they contain the halogen. So as per alphabetical order, chlorine comes first. So we'll start with these two groups. So N5, because there are two similar groups, one is attached at the end and one is attached at the fifth position. Hence N5 bis 4 chlorophenyl 3 propan 2YL imino, not count as the amino. It is imino because there is a double bond. Hence it is imino 3 5 dihydro, as there was a double bond, but there is electrons are missing on the third position and the fifth position. Hence we consider 3 5 dihydrophenazine. So the name of the ring is phenazine. So in phenazine there is a double bond at 3 4 and at this 4 5, but the electrons are missing at 3 and 5. Hence we write down the name as 3 5 dihydrophenazine 2 amine. So this is what the IPC nomenclature of the clofazimine. Although the clofazimine is classified as a secondary drug for the treatment of leprosy and commonly used as a component of multi-drug therapy, so this drug, clofazimine drug, is given in combination with the other drugs to overcome the resistance of that particular drug. Clofazimine was first used to treat advanced leprosy unresponsive to dapsone or streptomycin in year 1966. So this drug, as I have told you, that this drug is given as a multi-drug therapy. So why it is given as a multi-drug therapy? Because those patients who develop resistance against the dapsone, so to treat the leprosy, this clofazimine is given with the dapsone or with the streptomycin to treat the leprosy. The chemical, a phenazine derivative, is a water soluble dye. A phenazine derivative, it is a water insoluble dye that lead to pigmentation of the skin. In addition, discoloration of the faces, eyelid lining, sputum, sweat, tears, and urine it is seen. So this is what some side effects of this clofazimine. So this kind of effects was is observed when the clofazimine drug is given to the patient for the treatment of the leprosy. This clofazimine increases prostaglandin synthesis and the generation of antimicrobial reactive oxidants from neutrophils, which can play a role in the anti-leprosy effect. So this is what the mechanism of action of the clofazimine. That is, this drug increases prostaglandin synthesis and generates some reactive oxidative radicals that that reactive oxidative radicals that are used against the bacteria, that is the mycobacterium leprae. Regarding the SAR studies, the imino group appears to be essential, with activity increased when the imino group is substituted with alkyl or cycloalkyl group. So over here, this amino group should be substituted with alkyl or cycloalkyl group to produce the desirable effect. Halogen substitution on the para position of the two phenyl enhance activity but are not essential to the activity. So these halogens are essential for the activity. The following order of activity has been reported, that is bromine is more significant as compared to chlorine, then methyl, then ethoxy, then hydrogen or fluorine. These are the metabolites of the clofazimine. Again, uh, these clofazimine undergo hydroxylation reaction to produce this metabolite, and after hydroxylation, it undergo phase 2 metabolism to form the glucuronide conjugation reaction. Again, the clofazimine undergo hydroxylation reaction by removing a halogen, that is a hydroxylic dehalogenation reaction, and after the hydroxylic dehalogenation reaction, it undergo phase 2 metabolism to produce a sulfate conjugation product. So these are the metabolites of the clofazimine. And the last drug for the treatment of the leprosy is rifampin. So as we have already discussed this drug in the previous lecture, that is in the treatment of the tuberculosis. So rifampin, it is an anti-tuberculin drug. Its action against Mycobacterium leprae, it is parallel to those effects reported for the Mycobacterium tuberculosis. Today, rifampin is considered to be an effective anti-leprosy agent when used in combination with the sulfones. So this is what all about the leprosy.